Our goal in training our graduate students is that we teach them how to ask questions. So a student comes into my lab interested in the broad question of how cells maintain genetic stability, and they learn to ask specific biological questions that take a genetics, molecular biology, and cell biology approach to this problem.
So I'm going to take this plate of yeast and I'm going to make a copy onto different kinds of plates. So one is a drug plate and another one is a plate that has a chemical that helps us look for fitness. So if the cells are sick or if they're healthy.
My lab studies a fundamental form of cancer research. We're interested in the process of cell division, and in particular, how cells maintain their genome, that is, their DNA, accurately. So if you damage cells, a perfect example of that is smoking, which damages the DNA of cells. If you have lost the ability to repair that DNA damage appropriately, the damage that you've acquired may accumulate, create mutations that result in cancer. And these are, again, live cells. They're growing and being imaged on the microscope. And you can see that the cells are growing and dividing, and they divide from 1 into 2 by medial fission. And most of the cells look like they're doing this in a very regular fashion. But if you look at this cell right here, it's taking a long time to divide, and when it finally divides, it looks like it's dividing into 3 instead of just 2 nuclei. There it goes. It's going to be a highly abnormal division. These cells are all mutant. They're missing a particular protein involved in chromosome structure. What's interesting is that loss of this same gene in humans is associated with metastasis in breast cancer. So even though we have an organism here that doesn't get cancer, we can study the function of a cancer-associated gene in this organism by looking at this sort of phenotyping. So understanding the mechanisms that cells use to maintain normal genome stability gives us insights into what happens and goes wrong in cancer cells. I joined USC in 2004. At the time, the strategic plan had just been developed, and this building, Irani Hall, was just being built, which stressed a commitment to basic life sciences on this campus. And it was very exciting to be part of creating something and building a program and a new center of excellence in this research. I love having students of all levels in my laboratory, from undergraduate to graduate, as well as my postdoctoral scholars. And I wanted to be part of an environment where there was a combination of research and teaching.